So there was an emergency meeting in Malacanang, and that was December 26. At kami inimbitahan ng Department of Health. And then nandoon si President Duterte doon sa Malacanang, and he asked Secretary Duque na, kaya ba natin yan? Mayroon ba tayong kakayahan mag-sequence niyan? Yung sinasabi ng WHO. So pinatawag ako doon ni Secretary Duque at sinagot ko naman si President and the members of the cabinet na actually within the University of the Philippine System, we have an existing Philippine Genome Center and we have all this next generation equipment and we're ready to do the analysis.
Magandang araw mga kapwa-siyentista at welcome sa BTS o ang Behind the Science podcast kung saan kakausapin natin ang mga scientists from UP Diliman College of Science and UP Resilience Institute. Dito sa podcast ay pag-uusapan natin hindi lang ang mga resulta ng pag-aaral nila, kundi pati na rin ang mga behind the scenes or behind the science stories. Simula sa kung paano nila ito naisipan gawin hanggang sa na-publish na nila ito. This podcast is co-presented by the UP Diliman College of Science and UP Resilience Institute. Ang episode natin ngayon ay all about COVID and we have a very special guest. Isa lang naman siya sa mga instrumental na tao na nag-handle ng pandemic. So siya ay nagtapos ng BS Fisheries sa UP Visayas and BS Molecular Biology sa Nagoya University sa Japan. At nagtapos din siya ng Master of Medical Science in Physiology and PhD in Physiology sa Osaka University sa Japan. Ngayon, siya ay professor sa UP National Institute of Molecular Biology and Biotechnology and also a principal investigator of the Laboratory of Molecular and Cell Biology ng UP-NIMBB. Noong 2008, she was awarded the UP Diliman Centennial Gawad Chancellor para sa Natatanging Guro Award. At noong 2017, she was appointed as the Executive Director of the Philippine Genome Center. Noong 2022, she was also awarded the Presidential Lingkod Bayan Award by the Civil Service Commission and also the Presidential Recognition for the UP-PGC for COVID-19 response awarded by the Office of the President. So without further ado, let's all welcome Dr. Cynthia Saloma.
So magandang umaga, Paul, at sa lahat ng ating tagapakinig dito sa BTS program ninyo.
Maraming salamat po ma'am sa pag-accept ng aming invitation. And as usual, bago natin pag-usapan yung COVID, pag-usapan muna natin yung paano po kayo naging scientist hanggang yung current position nyo na po. So, can you share with us ma'am, when did you realize na gusto nyong maging scientist?
So actually, Paul, I became a scientist by accident. So when I was a young child, I was privileged. No, I was privileged to get enrolled at the School for Exceptional Children in Iloilo. So yung lahat itong mga bata na mga nasa elementary school, kumuha ng exam. And then we entered this School for Exceptional Children. So merong mga mentally gifted, meron din tayong mga batang may hearing impairedness or iba-ibang challenges. So it was a very, very good school because it allowed us the opportunity to explore art, science, at the same time, of course, the special giftedness of other children, no? Special, and also children of special needs. So noon, lahat kami, nung graduate kami ng elementary, pag pumasok ka ng UP High School sa Iloilo, automatic para kang valedictorian kasi binigay para kaming mga entrance scholars lahat-lahat. So it was a nice thing kasi palagay ko halos lahat kami hyperactive at saka ini-explore talaga ng school din yung aming artistic tendencies. So nang nasa UP ako, gusto ko talaga maging abogada or I really wanted to become a lawyer. So I was also interested in physics as well as in mathematics and in history and so, of course in economics. So ang dami kong hilig, hindi ko na alam tuloy kung ano kong gusto kong gawin, pero in my estimation, parang mas gusto ko talaga pumunta into law. Tapos, nung kumuha ko na exam, nakapasa nga ako nito sa College of Fisheries entrance exam. So, parang, ito yung exam na kinuha ko na parang walang tulog ba? JS prom kami tapos 4 a.m. ko umuwi tapos 7 a.m. may exam. So, tapos nakuha ako. There were 5 students, every region nakuha. Sabi ko parang providential. So, tinanggap ko nga yung scholarship. Pinag-isipan ko talaga ng mabuti kung tatanggapin ko nga yung College of Fisheries na scholarship. So, pumunta nga ako sa College of Fisheries and then I realized, within the College of Fisheries, marami ka namang makuhang pursuit. Di ba? You can even go to medicine, you can go into law, you can go into genetics. So, kahit nasa college na ako at nasa College of Fisheries na ako, kumuha pa rin ako ng mga subjects sa economics, accounting, at saka sa poli-sci pa rin. Kasi I really wanted to go into the College of Law. Now, noong graduating na ako, kumuha ako ng exam sa Mombusyo. And I was, no, not graduating. I finished pala in 3.5 years. So, kumuha ako ng exam sa Mumbug ako siya, sa Japanese Ministry of Education exam doon sa Japanese Embassy. And I took the exam in Cebu. Ang hirap ng exam kasi parang sinabihan ako, the exam will be tomorrow, tapos I have to go to and fly to Cebu and to make many arrangements. So, and if I can remember correctly, Paul, I was the last one to board the plane. I was so late, and my family did not want me to really go to Japan and get separated. Nevertheless, kumuha nga ako ng exam. Tapos pagbalik ko sa bahay namin, sinabihan ko tatay ko, ay, don't worry, hindi na ako makakapasa doon kasi ang hirap nga ng exam. Iba yung exam ng Mombusyo compared sa UPCAT at sa iba nating exams.
Ay,
Kasi
ito po ba yung ano, yung mixed?
oo. For undergrad ha, kasi undergrad ako at saka tayo, 'di ba? Wala naman 4 years lang ang high school natin. So this was an undergraduate exam. I was in third year college na. So nung kumuha ko ng exam, ibang klase yung exam nila sa chemistry, ibang klase yung isa. Tapos it was really without any preparation kasi the day before lang ako sinabihan na kumuha lang ako ng exam sa Cebu. So naghanap ako pambili ng ticket, and then iyak ako ng iyak sa bahay kasi ayaw ng tatay kong pumunta ako magpunta ng Japan. Sabi ko, di ba nga ako nakapasa Sabi ko. Anyway, nang bumalik na ako sa bahay namin, and this was really, really funny, sinabihan ko na yung father ko na, Tay, don't worry, I'm not going to make it kasi the exam was so hard. At saka parang, you have to fill in the blanks sa mga periodic table, mga ganon. Grabe ang exam. Tapos mga reduction ng mga dry cell at kung ano-ano pang mga exam. So it was hard and the math exam was also hard. Anyway, nakuha ako as I will be recommended for the Japan scholarship na sa MEX. Akala ko, Paul, sigurado na yun. Pinamalita ako na sa buong mundo na pupunta na ako ng Japan. Hindi pa pala sigurado yun. Meron pa palang next screening. Mabuti na lang nakuha ako by January. So anyway, katawa
May
na.
interview pa po yan
Oo,
pagkatapos.
oo. Actually ako, wala na akong interview kasi nasa Iloilo ako, di ba? So I was not
Ah.
interviewed at all. I was recommended to Japan. Akala ko talaga 100% sigurado na yun. Mabuti na lang nakapasa ako, no? Otherwise, nakakahiya pala na binalita ko na sa buong mundo, I'm going to Japan. So anyway, the day after my undergraduate graduation, di ba natapos ako maaga, 3.5 years. So anyway, nung nag-graduate na ako sa College of Fisheries, ang plano ko talaga mag-masters na sa Japan. So nandun, pero of course, nung napasahan ko sa Mombusyo, undergrad, hindi pa ako qualified sa masters kasi wala pa, third year pa lang ako noon, nang kumuha ako ng exam. And then when I went to napunta na ako ng Japan, kumuha ako ng Japanese language, ang gagaling ng mga kaklase ko kasi yung iba marunong na sila mag-Japanese. Ako zero talaga, zero as in zero. Of course, we studied Spanish during that time. So, can you imagine, Paul, the first time I was there, tinanong kami ng teacher namin in Japanese, kasi the teacher will never speak in English. Tinanong ako, tapos sinabi ko sa sensei, sabi ko, si sensei, pero it reminded me of my Spanish class na I had to be taught in a foreign language. So nevertheless, nagpunta na nga ako doon. Tapos I wanted to go to grad school na nga. So I was thinking about genetics or nutrition, di ba? Fish nutrition or fish genetics. Kasi nag-fisheries nga ako. Hindi pala ako pwede kasi nga undergrad na ako. Tapos sabi nila, just take any, at saka syempre wala naman akong work experience. No? So, I was asked na just take any undergraduate course at saka na lang ako magpunta sa grad school. So, when I was thinking about it, I decided to probably, sabi ko, mas maganda siguro mag-molecular biology ako, di ba? Kasi genetic engineering. So, tinulungan naman ako ng teacher na Japanese at kumuha nga ako ng exam sa Nagoya University, which is have a very, very good reputation on molecular biology. At saka maraming mga Nobel Prize winners ng Japan of the 21st century are really coming from Nagoya University. So I had a very good time in Nagoya University. So I studied molecular biology. And the model system I studied was the mouse. Pero since ang teacher ko naman, alam niyo naman na fisheries ang graduate ko, nag-aral din ako ng Medaka as a model system. So sa developmental biology where the lab I was I joined in, we usually study model organisms to model human diseases. So yung lab namin, we were studying the mouse, but at the same time, of course, I deep in my brain na eventually I'm going to study the fish na as a model system. So, yung laboratory namin nag-move na so Paul, during that time, I studied knockout mice, no? So, akala ko nga naman, hindi ako mag-medical doctor kasi parang takot ako sa mga blood, etc., etc. That's why I wanted to go into law. But nevertheless, maganda din naman yung science, the discovery and the challenges that you go through. So, I went with my professor when he moved to Osaka University, and I took an exam in the medical school. So we were the first students of the newest building on campus. It was a very new building of the medical school of Nagoya, of Osaka University. And it's a very, very modern building. And we were the first students. Maganda yung campus. At saka isa siya special curriculum. Parang yung first year namin, puros medical sciences. Tapos yung second year namin, puro na laboratory na. So parang a little bit different from the usual Japanese curriculum. So but I moved on from medical school and then college. Pareho pa rin ang advisor ko. So I was working with knockout mice. Di ba, Paul, during that time, may mga stem cells na or embryonic stem cells. And ang pinakasimpleng explanation is, di ba, we have proto-oncogenes or oncogenes which cause cancer. And then when you knock out the gene, yun pala, many of them can also cause embryonic lethality. So ibig sabihin, kailangan din natin itong mga genes for cellular division and the proliferation of tissues at saka organ formation. And yet, kung deregulated sila, magkakakos din sila ng cancer. So that was what I was studying in Japan. So parang nakalimutan ko na yung original desire ko na mag-law kasi na-embed na ako doon sa wonders of science and the use of model organisms and studying the mechanisms of how organs are formed, how the brains are formed. So when I came back to the Philippines, I got married pala. In the interim, I got married. I met my husband in Japan. Of course, you know everybody that is a Dr. Cesar Saloma. When I was during his postdoc, we met. So anyway,
Ah,
to make the story short, we got married, and I still went on to go to grad school.
sa Japan pa kayo nagpakasal,
Oo.
hindi pa sa
Ay hindi,
Pilipinas?
sa Molo Church, siyempre sa hometown palagi ng lady yan. So we got married in Iloilo City in Molo Church about 2 weeks before my second undergraduate graduation. And then he joined me for 6 months as a visiting professor while I was in graduate school in the medical school of Osaka University. So he also did his PhD research in Osaka University. So we have also common friends in Osaka University, but we met in Nagoya University during his visit with to his friend na nag-graduate din doon sa Osaka. So binisitahan niya si Dr. Tony Fernandez sa Nagoya parang International Center. And that was one we met, and that is to shorten the story, nagkatuluyan nga kami. So actually, Japan is a special place for us because that is that is where we met. Okay, so pagbalik ko na ng UP, I had to go back kasi, di ba, I already got married. And during the time, MBP was still not a national institute. It was still just a program, MBBP. Eventually, we exerted effort to make it as a national institute, and I started a laboratory of laboratory mites. No? So we did not have lots of grants during the time, Paul. Ibang klase, yung parang research money namin, parang 50,000 a year lang. It was a little bit very, very challenging for us to pursue research. At syempre, yung aming laboratory, hindi molecular biology grade, di ba? Kasi originally for the College of Fisheries siya. So, but we made do with that. And we always dreamt na siguro when there is we intended na sana magkaroon kami ng totoong building talaga. So we had that opportunity. We went to Malacanang, and it was the time of President Gloria Macapagal Arroyo, and that is how we started the National Science Complex. And we built this MBB building where I am now na para siyang R&A loop kasi may curve. So ang tawag namin parang R&A loop building. So that's my story while I'm now in science. So sometimes, when opportunities arise, we just make we try to grab these opportunities that come our way.
Taga Iloilo pala si ma'am. So fun fact lang,
Oo.
Ilonggo din po ako, ma'am.
Hello Paul.
And
Oo, oo.
so
Ahead.
ma'am, magtatagalog pa ba tayo dito?
Pwede man mag-Ilonggo. Katawa. During COVID, Paul, there was a time that we had to go to do a town hall meeting sa Region 6. Di ba? That was a time na tumaas talaga yung mga Delta cases sa Iloilo City. So nung nag-town hall meeting kami with the governors and the mayors and of course the DOH officials, in the end, alam mo, nag-Ilonggo na lang ako, no? Just to tell them na, we have an Ilongga here. We have the best interests of our province and our region at heart. So when I was talking about yung mga COVID and the spread of, mga variants, I was doing it in Ilonggo. And then there's another resource person na taga Cagayan de Oro naman siya. So when we did our town hall meeting sa Cagayan de Oro, nagbisaya naman siya. So I think it was a good time. Now we try to connect with the regions that are close to our hearts. No?
Ayun. So since nabanggit niyo na po ma'am yung COVID and also yung town hall meeting niyo sa Iloilo. Pwede niyo po bang ma-share sa amin yung mga
Behind the
kasi
scenes? Oo.
kasi instrumental yung PGC dun sa COVID policies during the pandemic. So can you share po yung mga studies na ginawa nyo during that time and how it influenced yung mga
Okay.
COVID policies?
Oo. So Paul, when we started the Philippine Genome Center in 2012, no, it was really in anticipation of a pandemic. Hindi namin alam magkakaroon ng pandemic, but we really anticipated that there was going to be a pandemic because there were some people who were actually dissuading us from investing in a next generation sequencing machines because ang mabilis ang obsolescence ng mga machines na ito. But we realized na if we don't have a tangible equipment within us, and at the same time, if we want to train people and to do our own analysis, hindi pwedeng i-outsource mo lang. And also because, actually, very important dito si Dr. Amelia Guevara, kasi paano kung magkakaroon ng pandemic and then the borders are closed? We don't have a way by which we can understand as well as identify the causative organism, di ba? So dahil na rin sa experience sa SARS and all the other pandemics, we decided na in addition to providing our scientists locally with a service facility na hindi na nila ipadala yung mga samples nila abroad, kailangan magkakaroon din tayo ng mga sequencing equipment para kung may mag-pandemic at nag-close ang borders, magkakaroon din tayo ng kakayahan na malaman natin kung ano yung mga pathogens na ito. So those were some of the reasons why the DOST PCIRD gave us the initial money for the start of the UP Philippine Genome Center. Tapos, nadagdagan pa yung ating equipment doon sa project din ni Dr. Eva Cochonco de la Paz tungkol sa yung CHED naman, dinagdagan pa tayo ng mas expensive at mas powerful equipment in 2017. No? Ito yung mga sa CHED Picari project. And we really wondered kasi, syempre, this is an awful lot of responsibility on our end na dapat yung mga equipment ng investments ng ating pamahalaan at ng ating taxpayers should be very meaningful. No? So naisip namin, magagamit kaya itong mga equipment na ito sa NGS kasi syempre ito sa new technology at saka parang ang ating mga scientists nagsisimula pa lang sa kanilang mga research sa whole genome sequencing, etc. etc. So nevertheless, nagkaroon pala itong mga variants of concern. Yung lumabas yung UK variant, you remember that was the UK variant. So actually in the beginning of the pandemic, so that was January, we already prepared ourselves dito sa PGC in case we will be called upon to do the sequencing. Pero in the beginning, hindi naman kami kinontak na mag-sequence. But we actually got involved with validation of the kit ni Dr. Raul Vistura, yung ano no, yung MTech na kit. So we were able to do in that during that time we actually did a sequencing, pero ito yung sequencing na we sequence everything. So it's called metagenomic sequencing. So we were already involved, and we were just waiting for the government to also ask us if the need arises. So nagkaroon ng mga variants ng mga D614G when we opened our borders. Pero importante, Paul, na in addition to our role in the sequencing of the COVID genome, actually because we are molecular biologists at the Philippine Genome Center at the MBB in UP Diliman, we realized that our expertise in molecular biology was really, really very important in order to increase our nation's capacity for testing, di ba? RT-PCR. So, ang ginawa namin dito sa UP, bago kami nag-genome sequencing actually, is really to create an online platform as well as a hands-on training program together with the National Institutes of Health para matulungan natin yung RITM at ang ating Department of Health na maparami pa yung mga licensed labs. So, sa totoo lang, ang ating, through the University of the Philippines, OVPAA, nagbigay sa atin ng pera. Ang ating mga scientists dito sa College of Science at saka sa National Institutes of Health in UP Manila, together with, of course, our partners in the Department of Health, we were instrumental in the licensing of more than 200 labs. No? So to spread our nation's capacity for testing kasi di ba ang tagal-tagal ng ating turnaround time sa RT-PCR testing. So a lot of them were trained by us, either in the beginning hands-on, kahit yung borders, and then also online. And when Visayas and Mindanao, PGC Visayas and Mindanao were already up and running, they were also helping train people on molecular biology techniques. So that was one. So we were on standby. So we were instrumental in training more laboratories and more medtechs, and also pathologists on understanding what is RT-PCR, how do you read these mga city values sa testing. So yun ang first phase. Second, nagkaroon pala it really created a stir kasi naglumabas yung very aggressive form of COVID. Ito yung ng June 2020, lumabas ang D614G na mayroong additional mutations in Europe. And then the entire world community was called upon na kailangan magkaroon daw ng genomic biosurveillance kasi lumalabas itong UK variant, yung B.1.1.7. So there was an emergency meeting in Malacanang, and that was December 26. At kami inimbitahan ng Department of Health. And then nandoon si President Duterte doon sa Malacanang, and he asked Secretary Duque na, kaya ba natin yan? Mayroon ba tayong kakayahan mag-sequence niyan? Yung sinasabi ng WHO. So pinatawag ako doon ni Secretary Duque at sinagot ko naman si President and the members of the cabinet na actually within the University of the Philippines system, we have an existing Philippine Genome Center and we have all this next generation equipment and we're ready to do the analysis. Nako, within 24 hours, dumating na sa amin yung mga samples galing sa airport, no? Mga galing sa airport with coordination with, di ba yung mga nagsaswab dati, mga taga Philippine Coast Guard. So, within 24 hours, nagpadala na sila sa amin. Pero syempre, Paul, bago yan, meron na kaming funded projects through the DOST na just in case kailangan namin ito at So, since wala pa kami tinawag dati ng DOH, gumawa talaga kami ng project para sa genomic biosurveillance. Ang ating project leader nito si Dr. Ben Maralit. So, kung baga, bago kami pinatawag sa Malacanang, mayroon na kaming existing funded project ng PCHRD na mag-track ng mga COVID variants. So, nang pinatawag na kami ng Malacanang, kung di ba ready na kaming reagents, no? Ready na. Otherwise, kahit sabihin sa amin, mag-sequence kayo ngayon, di ba the entire world community was really looking for all these sequencing reagents. Kung nangyari yon, di 3 buwan pa hihintayin namin, mabuti na lang naka-standby na yung mga reagents to our PCHRD grant. So, nagawa namin yung sequence and our first publication, and it was a really big news, is na-detect natin sa Mountain Province na meron palang UK variant doon, yung B.1.1.7. So that was very, very important. And we had to report to IATF almost on a weekly basis kung ano yung nakikita namin. So ako, ang aking duty doon is we do the sequencing in coordination with the Epidemiology Bureau. And of course, syempre, dadalhin ito sa Diliman, isi-sequence namin. So yung aming mga scientists dito at ang ating mga nagsisequence at nag-bioinformatics poll during that time, para namin silang nasa loob sila ng building, ayaw namin silang magkasakit, 'di ba? Kasi kung magkasakit sila, di syempre mapuputol yung ating analysis. So that was very, very important na i-house namin sila doon sa NISMED at saka sa mga dormitories para na kung baga naka-lockdown sila, because it was very, very important that we give our government a timely answer to the questions they wanted to ask, particularly on the spread of the variants and the mutations that we are looking at in the field. So that was also the reason na nakita nga natin yung P.3 Theta variant, you remember? There was a lot of cases pala na nangyayari sa Region 7, and we were contacted and they sent to us through the plane, no, by plane. Pinadala nila sa amin yung samples nila, and when we were sequencing it, para siyang Brazil variant, pero nang we were looking at it, we were looking at something very new. Iba yung
Gaano
molecular.
po kadami ay sorry po.
Oo.
Gaano po kadami yung mga samples na ina-analyze niyo during that
Oo.
time?
During the time, so we started, let's say, 100, tapos 350, tapos 700, up to 1,500 yung at a single time. So, depende sa dami. Kasi marami naman kami, Paul, ng equipment dito sa PGC. Pag kaunti lang, let's say 24 to 48 lang, we use a MySeq machine, di ba? Kung nalala mo dati, Paul, na meron tayo kung minsan mga semen na may mga suspected Delta variant sila, and we need to give them an answer within 24 hours or 48 hours. So for that one, we use a machine na iba naman. So mayroon kami iba-ibang kits at iba-ibang machines. So depending on the samples that were given to us, we adjusted kung ano ang platform. But most of the time, our workhorse talaga was the Nova 66000. And in the region, in Southeast Asia, very few lang ang mga laboratories na mayroon ng ganong machine. No? So we were very lucky that we had that equipment called Nova 66000. It was our workhorse for COVID sequencing. And many of the labs around the world, manotice nyo yung kanilang sequencing sa 750, 750. The moment we say 750, most likely they were running it in a Illumina Nova 66000. So we were sequencing from kung minsan 24 kasi mabilisan, kung minsan 350. And for that, we were using a Next 6 500. And then, normally, 750 ang aming machine, ang aming pinapa-sequence. And eventually, Paul, what happened was we needed to do also the sequencing in Visayas and Mindanao, and we had to train them on this one. We were also lucky that we were able to provide the necessary expertise in the regions. And we were very fortunate na yung ating Bayanihan funds were able to fund the expansion of PGC to Visayas and Mindanao, together, of course, with built-in equipment from the OSTPC HRD. So at the peak of the pandemic, we were sequencing from 750 to 1,500 every week at that time. So yung story naman doon sa B117, and that is of course one of the first publications we have, was we detected these in, number one, we detected these in the Mountain Province. The other one is we actually detected these among travelers in Dubai. You remember the travelers in Dubai. And in Dubai at that time, they were not sequencing. They were not doing active genomic surveillance. And of course, there was a very brisk tourism there. We had some Filipinos from the National Capital Region who visited there. And when they were returning to the country and they were swabbed in our borders, we realized na meron pala silang B117. So that was one. And then, of course, we also detected P.3 in Region 7, as well as, of course, in the National Capital Region. And we were a little bit concerned kasi parang wala siya sa ibang lugar. Nasa Pilipinas, kumbaga, nag-evolve ang mutation na yon. So, we had an emergency meeting with the DOH and other LGUs looking at P.3 or the Theta variant. So it was a variant of interest at that time because of some concerning mutations that we find in the spike protein. Mabuti naman na hindi naman nag-spread masyado. So, nag-evolve tayo, Paul, with the rise of cases. So yung una, yung UK. And then we detected also a lot of cases in Pasay. Remember yung yung African ba, no? South African variant, no? So it also brought to us the significance and of certain areas in our country where we have really a lot of movement. So, for example, our areas around the airport is really the place where a lot of our visitors come and also live. So, these are some of the vulnerable sites that we really have to look into. So, those were also incorporated as part of the policy of the IATF, no, to look into these areas kasi nakita natin unique among many countries around the world. Marami tayo sa Pilipinas ng B, no, yung variant na yon. So, ang alpha, ang beta variant, marami sa Pilipinas na na-sequence natin, particularly around the area in the airport. No? So, and we then, of course, during the time we detected the local variant, the Theta variant, that was actually had some concerning mutations because may mga mutations are very similar to the beta variant also na parang immune evasive. Kasi during the time, akala ng mga tao pag may vaccination ka, okay ka na. When in fact, vaccination does not really prevent you from getting infected, but it prevents you from developing the severity of the symptoms. So, yun yung
Ay,
ating
oo nga
mga
po.
story.
Na-COVID din ako nun eh. After nung first vaccine ko, na-COVID ako.
Oo. So, nakita natin po na, it was also a testimony, itong ating ginawa during the COVID pandemic. It was also a testimony na kaya pala natin gumawa ng bioinformatics analysis locally, ha? So, we were talking about the very young people here at the Philippine Genome Center, many of them with background in molecular biology, in physics, in computer science, in statistics, and in math, na tayo mismo gumawa ng ating mga algorithms at saka ng ating mga dashboard na makikita natin yung spread at saka yung mutations ng mga variants na ito. So, many of these, of course, are hindi natin makikita just like that sa public, but these are information we shared with the Department of Health and with the IATF. No? So, kasi tinatanong nila kami palagi for updates on what are the variants, etc., etc. So, until now, Paul, we still do that. It's not probably broadcast so much, but we're still doing genomic biosurveillance on a weekly basis in Visayas, Mindanao, and in Luzon. Pero syempre, masyadong mababa na yung ating mga cases, but we still track these mutations. And unless naman masyadong concerning or it really um, uh, put to concerning in terms of our healthcare capacity, we just track them, and our Department of Health is keeping tabs on them, but we don't make so much broadcast about them kasi wala naman. Nakikita namin kaunting spread, may mutations, pero so far naman wala naman kami nakikita, for example, na increased cases sa hospitals, etc., etc. But we're still doing those things, even now.
Maganda pong story na, I mean, in a way, yung
Mm-mm.
PGC pala, naka-ready siya para
Mm-mm.
mag-respond sa pandemic.
That's right. Mm-mm.
So siguro po yung follow-up ko dun, at that time, kasi 'di ba usually may mga allocated naman na funding yung mga
Mm-mm.
iba-ibang agencies sa Pilipinas. So during that time, yung funding po ba na binigay sa inyo, sapat po siya to respond or naghanap pa ng ibang paraan para
Oo,
makakuha ng
challenging.
additional funds?
Challenging talaga siya, Paul, kasi in the beginning of the pandemic, nagte-testing pa lang kami via RT-PCR. 'Di ba it was a time with a lot of uncertainty. Tapos syempre we are a government agency. At saka during the time, many of the suppliers hindi pwede utang. Gusto nila palagi cash, 'di ba? Cash basis. So syempre sa atin, sa government, hindi naman tayo nagbabayad in cash. So during that time, and when we were starting the Philippine Genome Center as a testing facility, we really benefited from the MBB diaspora. We have so many alumni all over the world na nag-raise sila ng funds, ang MBB Foundation, nag-raise talaga sila ng funds para matulungan kami pambili ng kits, pambili ng mga costume ba yun, PPE ng mga ating mga researchers, tsaka para na rin magbili ng mga some necessary equipment sa PGC. So I think they were able to raise, our MBB Foundation was able to raise about 3 million pesos. And that was very, very important. Now, during the pandemic, it was really a challenge kasi syempre, hindi naman kami nababayaran ng PhilHealth, di ba? Ang later pa nagbayad yung PhilHealth, until now we are still actually asking PhilHealth for reimbursement. So, nako, ang dami-dami naming kailangan, reagents, etc., etc. So, we borrowed from a lot of laboratories, from friends, and then eventually, ang government natin, through the DOH, nagbigay na rin, nag-transfer ng funds na sa university. Probably a little bit meron talagang lag, no? May lull talaga. So, we had to be very creative and asking for donations and mga reagents and equipment. At mabuti naman, alam mo, from the Philippine, yung mga OECD, no? Yung Office of Civil Defense, binibigyan kami ng PPE at saka yung ating sa RITM, kumukuha kami ng sample sa DOH at saka yung atin yung mga in general, the public, ang dami nilang tinulong din sa PGC, including yung mga supplies namin. Ang dami talaga. At saka daw, the scientific community banded together na pagkailangan namin yung mga ganitong-ganitong kits o tips, pinapahiram kami or nagdo-donate na lang. So eventually, nabigyan naman kami ng pera ng DOH, Bayanihan Funds, at saka nakabili na tayo ng maraming reagents. But it was always a moving battle kasi usually may lull talaga na 3 to 6 months that we have to find the mechanism by which we can still continue testing even if hindi pa dumarating sa amin yung funds. Pero eventually, na-work out na rin namin. So we just anticipated na may lull na 6 months and we need buy reagents, equipment, etc., etc. So much so, Paul, na towards the end of the pandemic, ang dami nga naming mga kits and reagents, ang baba-baba naman din ng cases natin. But of course, ganun talaga ang dynamics nitong pandemic na para tayong we had to rush and overcome some of the challenges, particularly in the logistics and in the supply chain.
Ay ma'am, curious pala ako kasi during that time, 'di ba may lumalabas na mga conspiracy theories na
Oo.
sinadya daw 'yan ng China. May
Oo. Ako
may
naman,
basis ba yung mga gano'n?
palagay ko hindi naman totoo yung, hindi naman, I don't think it was, in my professional judgment, I don't think it was an intended effort. There are a lot of possibilities. It could be an unintended laboratory leak, no? And so on and so forth. But I don't think it was intentional, if ever it happened. There are now a lot of possibilities na the possibility is high that it could have been a laboratory leak, but I don't think it was really intentional. And now, a lot of the world community, I think pandemic gave us a lot of lessons. You remember on paper, parang we are to be a united community, no? Pero in the end, parang sarili-sarili din naman, yun na-notice mo,
Hmm.
yung vaccine nila, inuna nila yung people nila, etc. etc. So, there were always challenges as far as equity is concerned. We are just very lucky sa ating na for some reason, our government was also very creative in finding for us vaccines. And na-notice mo, Paul, parang mabilis bumaba yung cases ng Philippines. Do you remember? Compared to other
Mm.
countries na supposed to be, bababa na yung cases nila, yung ating nearby in the region, mataas pa rin yung cases nila. And I don't think it was just a question na di tayo nagtitesting or anything. It's really because bumaba talaga yung cases natin, no? At saka even hospitalization rate. So, we have several theories regarding this matter. We don't have direct evidence, but we hope to do the crunching of the data. But it's probably also possible, Paul, na because we have many different kinds of vaccines and we did not focus on a single vaccine lang, I think it gave us a diversity of ang immune
Hmm.
system ba natin, no? As a general consequence, marami tayo kumbaga different kinds of antibodies generated. So, hindi kasi tayo nag-focus sa mRNA vaccine lang at sa ganito lang. Meron tayong attenuated vaccines, meron tayong subunit vaccines. And I think on the whole, and this is my professional opinion, I hopefully we can find some evidence later on when we crunch the data that I think this diversity of vaccines was very, very important for us. That our cases became fast, I think.
Ay ma'am, naalala ko pala, 'di ba parang may time 'yan na may nag-viral na may nagbigay ng statement na hindi mo na kailangan ng vaccine dahil yung virus mismo in a way yun yung magiging vaccine mo.
Oo,
Ano po
pero
yun?
in a way, ang mga viruses naman, usually ang design ng vaccines natin, yung classical design of vaccine is either a live or a dead live attenuated vaccine. So, pero they are in quantities or the potency is shortened, no? O is lowered so that by the time na yung totoo na talagang pupunta sa inyo, meron ka nang ma-memorize na ng cells mo na. ganito. So, I think the vaccines were really, really very helpful in reducing deaths and also, of course, in attenuating the severity of the illness. So, ito may mga iba tayong mga kaibigan na mga anti-vax sila. I think we cannot downplay the significance and the importance of the vaccines that were given to us. Of course, some iba potency, di ba? But I think the diversity of vaccines that were introduced to the country was very, very important na yung ating cases mababa, mabilis bumaba compared to other countries.
Tapos I think isa din po sa napansin ko, 'di ba, yung last na variant ay yung Omicron
Until now,
variant.
Omicron pa rin ang
Tapos
tawag. Oo. JN,
tapos after nun, parang wala na ulit news ng surge. Pero sa ibang bansa like US, may napapabalitaan pa rin ng mga surge.
yes, that's right, no? So so ngayon ang pinaka-circulating variant probably it can be as high as 75% of circulating variants sa US are JN.1 and their children are mga so JN.1. So we also detected JN.1 in the Philippines. There are some local cases na may medyo mataas ang JN.1, mataas ang cases nila pero we never experienced for example yung mataas ng surge or that our healthcare capacity is heavily challenged. Wala naman. Kaya on the whole, parang ang atin namang mga cases, mild na talaga. Magkasakit sila. They have some upper respiratory tract infections. And then after 2-3 days, mag-rest sila, okay naman sila. So I think managing our way out of the pandemic and even now, even if you have some cases, I think they're very, very manageable. And the severity of symptoms we do not are really, really super manageable and they are not as similar to the first cases when your hospitals were overflowing, etc., etc. So on the whole, sa Pilipinas, ang baba-baba pa rin ang kaso. Kaya wala na rin kami masyadong masisequence. Kaunti na lang yung nasisequence namin kasi kaunti na rin lang ang dumarating. At saka syempre yung nasisequence lang namin, Paul, yung mga naka-RT-PCR lang. Yung mga naka-antigen testing, hindi naman yan makarating sa amin. So yung mga nasa hospitals lang or sa mga testing centers na RT-PCR positive, yun ang nasisequence namin. So, on the whole, yung circulating variant, very similar naman to compare to other countries. Marami tayong mga GM.1 at so on. Pero, on the whole, hindi naman na yung ating healthcare capacity. Okay lang tayo and the symptoms are very manageable. No? But we, of course, as part of our pandemic preparedness and response, we are still monitoring the cases all over the country.
Since konti na lang po yung cases, is it safe to say na hindi na natin kailangan mag-worry sa COVID?
Oo, ganito. Sa palagay ko, hindi na siya necessary na we worry too much about COVID. I think it's very, very manageable. And the science and the medical establishment, mas marami na tayong alam on how to manage these cases. No? So, syempre, we still have some, from time to time, very low mortality, facilitated by probably because of, as you know, some people have some comorbidities and so on and so forth. Pero on the whole, I think it's a very, very manageable infection. And now the world is waiting for the next one. So there are a lot of preparation for the next pandemic. And so there are a lot of lessons that we can learn from the COVID and how it has been managed or was managed. And I think the world community is much better prepared for the next one.
Ayun. So, during the past, ilang years na ba? During the
2020
past maybe
yun, 3 to 4
3
years.
or 4
Oo,
years,
yes.
ano po yung may mako-consider po ba kayo ng mga memorable moments
Oh,
during
marami.
the pandemic? Yung mga good
Oo.
or bad na
Oo, sa mga
experiences na pwede niyo po ma-share?
oo, siyempre, Paul, grabe ang beginning ng COVID pandemic kasi parang we did not know na lockdown tayo sa UP, close na close, and then we had to think about ang PGC ba, should we go into testing or not? Di ba? You remember? Kasi syempre, everybody wanted to be safe from the virus and then we are going to do the testing here in the University of the Philippines on campus. No? So, of course, walang classes. No? So those are the things that we really have to think very hard kasi within our facility, may mga taong ayaw, may mga taong gusto. And then I realized tayo, we are molecular biologists and we are trained for this. And I think the country really needs us. So that was very, very important. So maganda yun, Paul. It was at a time na yung ating mga molecular biologists and alumni of MBB really came together, no, to help and to volunteer in the different testing facilities around the country. And I think it was one of those moments na nakita ng mga tao, important pala yung molecular biology as a discipline. It was it was one of the ways by which it opened the eyes of the world and of course our own countrymen na yung molecular testing pala very important. And molecular biology is a discipline, is a very important discipline. So that's one lesson yun. And number two is also it's very important that you have some form of preparedness. Sometimes, of course, within the PGC, we were prepared with the equipment and the talent and the bioinformatics analysis to respond to these things. So I think in many ways, it was also very, very important that we had the talent pool as well as the necessary equipment in place for us to be able to track and detect the mutations and the spread of the virus in our communities. And also, one of the things that were also very important to me was the fact that yung science communication, how you communicate science to the public, it has to be done in a manner that on the one hand, it has to be done very, very responsibly. On the one hand, it is important to convey the information, but at the same time, it should not be alarmist. So science communication and engagement with the public is something that we have to continuously learn, and but it was also very, very important that how we communicate the science to the public and how in a manner that is not honest, but at the same time not alarming. And the other thing that was also very, very important to us is for the first time in our generation and in our lives, we realized na the significance of science in action. Parang ganun. Not only were we able to, and our young people, our young scientists, were able to be involved in the detection of the virus, but also to understand its spread and also to educate the public about the things that we need to do. So, Paul, gusto ko pala ipaabot pala na how were we able to influence, for example, policy, di ba? Important yan. So actually, it was really based on science when we decided that we have to open our airports. Kasi nung towards the end of 2022, na, di ba, lumabas na yung Omicron, we realized na yung spread is not through the airports anymore. It was more community. So we showed that data to the IATF, and that was why the airport, the restrictions for travel was really open mga February. So some people were doing a lot of things, but it was really a very science-based decision because the data showed that the infection was spread more locally rather than through our international borders. That was one area na then all of a sudden the IATF declared na ang airports will be free for travelers, including tourists. No? So overall, itong pandemic provided us an opportunity, number one, to be of service to the nation and to show that science can actually contribute to societal problems, and also that the importance of science communication to our public and to our policymakers so that they can make informed decisions. And also that, coming together as a nation, both yung mga suporta na nakuha natin sa public, ang dami talaga mga volunteerism, everybody, yung mga nag-donate sa amin ng food at kung ano-ano pa, and the families of our researchers na syempre worried sa kanila but they had to be there in our building because they had to do all this analysis, and the many other collaborators who had all over the country who sent us to us samples generously. I think those were some of the experiences that will live with us for the rest of our lives.
Ayun, so nabanggit ni Ma'am yung Omicron and also yung role ng science communication. So side story na lang to. I think maganda din to i-share yung backstory or yung origins ng podcast na to. Kasi na yung podcast pala na to ay offshoot siya nung Marine and Earth Science Learning Hub. So yung channel na yon, ginawa ko siya nung January 2022.
Mm.
At ginawa ko siya dahil supposedly pupunta ako dito sa Japan nung December 2021. Pero 2
Uh-huh.
weeks before po nung flight ko, nag-issue ng travel ban yung Japan dahil nga dun sa Omicron variant.
That's right.
So
That's right.
ayun, sabi nung embassy, hindi nila alam kailan magre-resume yung flights. So wala akong magawa sa boarding house, naisipan ko gawin yung channel. And then
Uh-huh.
fast forward to now, eto na meron ng meron
So,
ng podcast.
eventually po, pumunta ka na rin ng Japan, Paul?
Yes po. Nung March 2022 po ako dumating dito.
Anong campus ka? Anong college ka? Anong university ka?
Sa Yokohama National University
Okay,
po.
okay.
And
Yes, yes.
under MEX scholarship
Scholarship,
din po
oo.
ako.
During the time kasi during our time hindi pa Mex ang name, Mombu siya tapos naging Mombugak siya na.
Opo.
Okay. So marami pala tayong mga stories about the COVID pandemic, ano?
Yes po. Ayun. So, almost 1 hour na rin tayo and I think kailangan na rin natin i-wrap up yung discussion. So, ma'am, before we let you go, do you have any advice to aspiring researchers in the field of molecular biology?
Oh, to your listeners and to our listeners, the young women and men who are contemplating to go on a career in science or even to go to molecular biology as a discipline, my message to you is science can be a very potent force for answering and responding to societal problems, and that has been brought forth during the pandemic. So, for our young people, I hope that if you feel that you have the aptitude and the talent to go into science, I completely support your decision, and I hope that our experience at the Philippine Genome Center at the NIMBB Diliman here at the College of Science will also inspire you to think about contributing to science and contributing to answers for societal problems. So now we can attack it at the level of detection, we can attack it at the level of diagnostics, we can attack this at the level of therapeutics or vaccine development. But in any rate, in any area you would like to go into, I hope you will do it with passion and with a very good and a very high sense of conviction. And I hope that many of you, of course, for those of you who are not in the country or will go out of the country, I hope many of you will also return to the country and be of contribution in training the next generation of scientists in our land. So, yun lang po masasabi ko po. Thank you very much for having me.
Also, ma'am, do you have any activities to promote? Activities from PGC or yung lab nyo?
Okay, so yung aming activity now ay my turn as Executive Director of the UP-PGC has ended in August after 6 years. So I am now the Program Director in Biodiversity, Ethnicity, and Forensics. So we are embarking in a program on conservation genomics, Paul. So what is this effort on conservation genomics? So we would like to sequence or at least make a draft sequence of all those, of many, not all probably, as much as we can of the endemic, endangered, and threatened species in the country. So, at the very least, within the Philippine Genome Center and in collaboration with our biodiversity team, we will have a repository of the genetic information of many of the um, organisms of the Philippines na hindi pa natin alam at nasi-sequence. No? So, and we also have a funding pala, and this is also an offshoot of the pandemic, where the UP-PGC and the University of the Philippines received funds from Congress to create the Philippine Genomic Information Resource Hub. So yung PGIRH is like the repository and also a mechanism where we can crunch data and deposit many of the genomic information in our country. So I think it's going to be launched with many collaborators soon. So para natin siyang gem bank ng Pilipinas, and it's going to be hosted at the Philippine Genome Center para yung mga ating mga information at mga data ng ating mga scientists. Some of them retire, some of them are not with us. Dapat na mailagay natin sa isang lugar at probably we could have a mechanism of data sharing or access, and at the same time the facilities within the PGC could be utilized by others who need a lot of computational tools to crunch big data, particularly in genomics. So yun, Paul, ang aking gustong ipaalam na we are embarking on this comparative genomics and saka conservation genomics project. And hopefully for the others who would like to get involved, we can create a consortium of people involved in biodiversity so that we can join or empower genomics together with conservation. Yun lang, Paul.
Ayun. So, thank you very much po, ma'am. And I hope may bago na naman na natutunan ang ating mga audience, especially yung mga behind the scenes na nangyayari sa anything we do in science. Madaming mga behind the scenes na nangyayari na hindi natin usually nababasa. So, hanggang dito na lang muna tayo ngayon. And see you sa next episode. Madami pa tayong pag-uusapan na ginagawa na research ng UP Diliman College of Science and UP Resilience Institute. So maraming salamat po, Dr. Saloma, for your time.
Maraming salamat din sa inyo and good luck to all your endeavors.